Hello to all that are watching. My name is Trevor DeGroot. I am a second year master's student at Bowling Green State University in Bowling Green, Ohio. My three advisors are Dr. Kevin McClooney, Dr. Helen Michaels, and Dr. Steve Hovick. And during this video, I am going to be presenting to you guys a summary of two research projects we have going on at BGSU. These are wetland mesocosm experiments, and they're aiming at sort of quantifying and studying the effects of hydrology and dredged sediment addition on wetland plants. So just a real quick background, the sort of why we're doing this in the first place. If you're not familiar with these images already, these are aerial and up close images of harmful algal blooms in Lake Erie. Obviously, they're not very aesthetically pleasing. They're very ugly. They create water that is pretty much you can't go into. You can't swim in it. You can't drink from it. And there's even some algae that produces toxins. So that can cause harmful effects to the health of people, the health of livestock, the health of anything really that lives in or around the lake. In addition to that, they cause millions of dollars in damages through cleanup fees, through making sure drinking water that comes from the lake is clean and pipes are cleaned out after. Overall, they're just pretty ugly and they cause a lot of damage. There was a particularly ugly one in 2014 that caused, a millions of dollars in damages and was pretty harmful to the area surrounding the lake. So in response to that, the governor of Ohio, Mike DeWine, instilled the creation of a overarching program, sort of in response to the algal blooms, but also in response to the smaller water crises that were happening in Ohio. So this program is called H2 Ohio, and it's aimed at cleaning up the water in Ohio, the water that exists both in your system, so in your house that goes through sewers, but also the water in lakes and streams as well. So it's sort of an overarching organization aimed at cleaning up all of the water in Ohio. And that happens through various ways. You can see here, this is an example of the targeted profile. So you have anything from reducing the phosphorus that comes from farm fields as a result of fertilizer or over fertilization or stuff like that that flows into waters. You have something like creating wetlands, which is what I will focus more on during this presentation. And then you have things that you more commonly think of as cleaning up water, like failing septic systems and preventing lead contamination. All of those are aimed at cleaning up the water in Ohio and surrounding Ohio, especially in the waterways. So, like I mentioned before, one aspect of H2 Ohio is the creation and restoration of wetlands. So, the reason why this is what this graphic demonstrates here, why wetlands are so important, and especially the creation of new ones, is that they filter water that flows from farm fields or flows from urban areas that is high in nutrients. And when that water, if it's not filtered and it flows all the way to the lake, it causes those harmful algal blooms and leads to, like I said, a lot of damage. So the creation of these wetlands allows the water to be filtered by flowing from streams and runoff, high energy flow into a wetland where it slows down. And a lot of those nutrients that are in that water either percolate down and settle as sediment, or they're taken up and incorporated into more stable forms that won't move by things like plants and microbes, both in the soil and in the water. So wetlands are, of course, also a very important ecosystem, but for the aspect of cleaning up Ohio water, they're vitally important anywhere. So, a big project of H2 Ohio has been the creation and building and restoration of wetlands around Ohio. So that's where I come in. Going into BGSU, I became interested in this whole H2 Ohio wetland restoration, wetland management project. And I was interested in plants in undergrad too. So luckily enough, I found the opportunity to become a research assistant on a H2 Ohio vegetation team that studies sort of how important plants are in this nutrient filtering function of wetlands. And in addition to that, through Dr. Michaels and Dr. McClooney, they submitted a grant that further tries to understand specifically what in plants allows them to better serve as nutrient filters and better allows them to hold on to nutrients. So that's sort of where I come in. Was my goal. My work is to study plants in the field, but also I'll touch on during this presentation in a mesocosm setting. Um, this is just this graph has a lot, or this figure has a lot of information on it. But what I would like you guys to look at is just look how many arrows are pointing into that plant there in the middle. This shows just how important they are in nutrient intake and nutrient storage and holding on to nutrients in their biomass. And not only that, but they're also responsible for taking in greenhouse gases like CO2. Um, and also, of course, pumping out oxygen, breathable oxygen for organisms that respire. In addition to creating a good root or a good below ground habitat for microorganisms and fungi and bacteria that further take in nutrients and convert them to forms that they'll incorporate into their biomass or more stable forms as well. So plants are sort of an over vegetation, or plants are a sort of a main shareholder in this whole nutrient cycling, nutrient retention, nutrient filtering function of wetlands. It's very easy, from a monetary and a management, especially when it comes to H2 Ohio viewpoint of looking at plants as just this sort of very important thing in nutrient cycling, but they're also vitally important for habitat, for hunting ground, for breeding grounds, especially in wetlands, as wetlands are a very rare habitat. They're declining every day. You have certain organisms that rely upon wetlands for feeding, for breeding, and then especially for habitat. So they're vitally important for that as well to a variety of organisms, including endangered species of waterfowl, of amphibians, and of course to more common things like insects or deer. This is just a picture here that demonstrates, all of the ecosystem services that plants provide. Ecosystem services are a service that an ecosystem will provide that relieve that to us humans, basically. So some sort of service. So you can see here, there's a variety of things. Some things I already touched on, like habitat and water quality improvement, but also things you might not think of, like a source of food and medicine or climate change mitigation. So plants are very important. They don't just sit there and photosynthesize and look pretty and stay still. They're doing a lot, and they're part of a lot of things when it comes to any ecosystem, but more specifically wetland ecosystems. Like for one of the projects for the hydrological experiment, we decided to focus on 12 species of native Ohio wetland plants. Well, 11 species and one sort of non-native that is a very common in wetlands. And these species were selected from work that H2 Ohio had done previously with servicing or surveying wetland vegetation. And these are the 12 species that we found that were not only native, but common in a variety of microhabitats within wetlands. So you have plants with a variety of morphologies, a variety of phenologies, so they flower at different times. And we hoped by subsetting these 12 species from the 100 plus species that exist in wetlands that we would be capturing a variety of different habitats, a variety of different conditions that each like, and a variety of different morphologies, and hopefully functional means to intake and hold on to nutrients. Just touching on a few here, you have in the top row, the second, you have Asclepias incarnata, which is swamp milkweed. That's a species that is more common to a typical plant architecture where you have a stem with leaves coming off the stem and then a flower on top. And then you have species in the bottom left corner, like Schonoplectus, which is soft stem bulrush, that are quite simply just a photosynthetic stem, which kind of serves as a leaf and a stem. And then you have a little kind of an odd looking flower at the end. So um, part of my research, I studied these species and I know a lot about them, and I've also almost I have a real affinity for these. So I just like to touch on those two as examples of the varying morphology. Now, for the dredge material experiment, as a result of it being sort of a sister project to the main hydrology HABRI experiment, we just didn't have enough young plants that were we when we put them into the experiment. We just didn't have enough to be able to have the same number of species in the dredge experiment. So we decided to take a subset of those six species. And these six species here are all very common at wetlands. You still have varying morphologies here, varying phenologies. You have the two annuals in here, which are also in the other 12 species. You have Bidens cernua, which is a nodding beggar stick, and you have a Echinocloa muricata, which is barnyard grass. So you have annuals, you have things that are more sedge-like or grass-like, and then you have things that are more typical plant-like. So again, a variety of morphology, hopefully a variety of different means to function as nutrient filters in wetlands. So now touching on the main two questions that we hope to answer with both of these projects, with the hydrology project and the dredge material project, both of them being Mesocosm projects. So the first question is, how do we design better wetlands? Knowing that wetlands are very important for nutrient filtering, nutrient sequestration, which is taking nutrients out of the water, out of the substrate. How do we design wetlands to function better as that? How do we design them better to function as habitat? Those two things tie closely into sort of what we're trying to answer with these projects. Like, how can we build and restore wetlands better in the future for H2 Ohio's purposes? And the second question is, given the different treatment levels and different conditions that we're hoping to mimic in this experiment, how do different species of plants respond to those conditions? And sort of what response and how does that tie in again with their nutrient filtering ability? So we hope to answer these two overarching questions. One a lot more broad, and then the other is a lot more specific. And I'll touch on those again and how we plan on answering them next. So when it comes to designing better wetlands, we sort of have two focuses. There's a variety, there's tons of different conditions and variables we can focus on wetlands, but as part of science, you can't really study everything at once. You sort of have to focus on things at a time and work your way towards understanding a whole ecosystem piece by piece. So for the HABRI, which is Harmful Algal Bloom Research Initiative funded project, which tied in with field work, we hoped to understand how differences in hydrology, so differences in soil moisture content, how that affects different species, and most importantly, how that affects the nutrient filtering ability of different species. So, do some species function better in certain hydrologies and then some don't, and do some function better in all of them? So that's one of our focuses is hydrology, sort of as a proxy to soil moisture, because while you might think wetlands are it's in their name. Their definition is that they're inundated for at least part of the year. There is a lot of variation in hydrology within wetlands. Within a single wetland, you can have areas that are very dry and you can have areas that are very wet. And then year to year variations, you can have a year where it's very, very wet all year. And then you can have a year where it's very, very dry all year. And then even within the year, you can have a year where in early May, it's super wet. There's a lot of rain. But then as you go on into the summer, the rain sort of stops, and then you have a wetland that's actually very dry when compared to other scenarios. So the other focus we have for designing better wetlands is dredge material amendment. So what I mean by dredge material amendment is that there is sort of an ongoing problem in Lake Erie because it's so shallow and because there's a constant flow of sediment into the lake. there's a lot of the floor of the lake is becoming filled with sediment, and it's really making the lake very shallow, so it's very hard for boats to navigate. That sediment has a lot of nutrients and other harmful things in it, like heavy metals and pesticides, for example, and that can really harm the lake. So for a while, since probably about the 60s and 70s, they've been taking excavators and digging, dredging that material out of the bottom of the lake and out of the bottom of the lake and streams and other things, but specifically the lake in this project. And they've just been putting it in areas, trying to clean it, trying to dewater it, and put that water back into the lake that comes out of the sediment. But eventually, there's not enough room to be able to continue to hold on to this because you have to continue to dredge because you're never going to, the flow of sediment into the lake is never going to stop. So part of um, this conservation work on Lake Erie is finding unique things, ways to use this dredge material that are beneficial. So, one example that a PI that is sort of leading this project is doing is using dredge material in agriculture, and it has a lot of positive effects for root morphology, for increasing the yield. So, what we hope to focus on by using dredge material amendment in wetlands is does it help plants establish themselves better? Does it help wetlands function better? So that way, it's sort of a twofold use in conservation, where not only are we um, taking dredge material out of the lake, which is good in itself, but we're also using it for a purpose that will lead less, that will help wetlands function better as nutrient filters. So it's sort of a twofold way. So in summary, the two focuses we have in designing better wetlands are to focus on hydrology and maybe focus on how to plant wetlands and restore wetlands, and when it comes to species preference on hydrology better, and then also does dredge material affect wetlands and affect the plant's nutrient filtering capabilities. So to answer a question one, which is designing better wetlands for nutrient filtering, and which sort of species are good at that, we have a process that goes on in these mesocosm experiments. So this picture here on the left, you have a plant that has been taken out of the tank. This is a picture of when we were harvesting the plant, and you can see that there's a lot of biomass there, and there's a lot of things going on there. So we will then separate that plant into the above and below ground tissues. This is just to get an understanding of where a plant is storing those nutrients that it has taken up, and sort of how can we understand where nutrients move within the plant. Um, and then we'll take that and we'll dry that down. We'll separate the tissues for the above ground material, and then we'll dry it down and put it into these bags in the oven, and then we'll weigh it, and that's to help us get biomass numbers because a lot of the time biomass is a very important indicator of basically how many nutrients the plant is taking up. It's also a very important trait for plants too, which I will touch on later. And then we'll take that dried material that has been through the oven, and then we'll grind it up. We'll put it in one of these little vials. It's just a subset of the total biomass. And then we'll send that to a lab, and the lab will get us the nutrient concentration. So, you know, milligrams per kilogram or whatever of nutrients in that vial. And then we'll be able to scale that back up with the biomass that we've already measured. And we'll be able to get the total amount of nutrients in the actual biomass. So it's not just it's not a concentration. It's not grams per milligram or milligrams per gram. It's an actual gram. It's a milligrams of nutrients that are in that tissue. So that way we can understand the nutrient stock, the actual amount of nutrients that's in that tissue. So that'll help us understand which plants are good at filtering, and that will help us to understand how to design better wetlands for nutrient filtering capabilities. And then to answer question two, which is how plants respond to the different conditions and how they adapt and function better in certain conditions as opposed to others is we're going to use traits. So overall, traits are an organism, a characteristic of an organism, whether it be physiological or morphological, that help it to survive and adapt in its environment, in its habitat. So some examples of traits that are commonly studied in plants, but that we'll focus on in these experiments here. are root architecture. So where a plant is putting its roots in the soil, how deep it is, and then how extensively branching and how many root hairs that root has, um, are very indicative of are very characteristic of certain areas. So if an area has a ton of water in the soil, the plant's not going to want to put its roots very deep because the soil down there is anoxic and it's basically it's hard to take up nutrients in that in those anoxic conditions. So root architecture is very indicative and it's very helpful. It would be very helpful to understand how root architecture changes when it comes to hydrology and dredge material. And then we have another example, which is biomass ratio. So um, obviously plants, they can move in some sense, but they can't get up and move somewhere else. So when conditions get poor or when they are in conditions that are sort of unfavorable, they might allocate biomass to different areas. So for example, they might send more biomass to their roots in certain conditions because they want to sort of stay like almost hibernate. And then when conditions become more favorable, they'll use all that biomass that they sent down to their roots and they'll allocate some more of it to the above ground. So when conditions become good, they can sort of sprout up again and photosynthesize and flower. So biomass ratios between roots and stem and different or roots and above ground, and then different tissue types like stem and leaves can be very indicative of how a plant is responding and what it's doing, how it's functioning in its environment. Now I'm going to outline some expectations that we have for the two different projects. In lieu of explaining very complex hypotheses, I just want to give some overarching hopes that we have for this experiment. So in the hydrology experiment funded by HABRI, we will have three different soil moisture levels. These represent our three different treatments. We will have a treatment that is hopefully um, achieves very high soil moisture, and the soil is very flooded. And then we will have a treatment that'll be medium soil moisture. This is our control. This is more typical to wetlands we see in the field when it comes to soil moisture. And then we have a final treatment that's very, very low soil moisture, where we are trying to mimic a drought for the last 4 weeks of the experiment. So, a wide variety of soil moistures, but we're hoping to understand, with the heterogeneity that happens in wetlands, how do plants function, and how do they manage to function in that. Um, so for expectations for when it comes to designing better wetlands, we will hope that certain species of plants, so you can see here that there are 6 different species of plants here. This is just an example. These plants may not function necessarily better in these soil moistures, but this just shows we would expect that different species of plants will function better as nutrient sequesterers and nutrient filterers in different soil moistures. So we hope to see a diversification of which plants function better in which soil moistures. So that way, they function better as nutrient filters. So you can see here that these two common nutrients that we expect them to uptake and put into their biomass are phosphorus and nitrogen, which those are contribute to harmful algal blooms in Lake Erie if they get into the water. So we hope that one expectation we have is we hope that different species will function better in different soil moistures because that's not really well understood yet and it's not really heavily studied either. The second aspect when it comes to traits and how plants respond to certain conditions, we hope that the different soil moistures, or we expect that the different soil moistures will lead to a difference in traits. So these are just two pictures that are mashed together. They're not even necessarily the same species, but they're just trying to show very simply that there's going to be differences in things like root architecture, which you can see here, for example, in the high soil moisture picture. I tried to create a picture that shows the roots will be more spread out because they're still trying to take in a lot of nutrients, but they don't want to go very deep because it's hard to take up nutrients when the water is very high and there's a lot of anoxia. So and then the other treatments look different when it comes to root tissue and above ground material too. So you see that we hope that there's going to be a diversification of traits that allow plants to survive in these different conditions, and that some species may not be able to survive in certain soil moistures or respond or function because they don't have the traits necessary that allow them to survive. So all in all, we expect that With the different soil moistures, there will be a diversification of traits that allow them in response to the different soil moistures, and allow the plants to survive. And in that, in turn, will affect the amount of nutrients they're able to take up in those certain conditions. And now, the expectations that we have for the hydrologic or the dredge material experiment are that with an increased amount of dredge material added to the soil in wetlands, which is depicted with this increasing in the size of this picture here, you can see that as you increase in dredge material, we hope that this will increase juvenile establishment of plants. So what establishment means is that they're able to, successfully shoot their roots out. They're able to, find themselves happy, and good to go when it comes to growing and producing biomass and taking up nutrients. So we hope that as you increase the dredge material, you increase this establishment. And what that establishment entails is that you will increase the biomass of the plants. So you will see an earlier increase in the accumulation of biomass as plants are establish themselves quicker than they do in soils that are not amended with dredge material. You will also see that there will be an increased growth rate. This is just tied in with that whole increased juvenile establishment. And then you also see, hopefully, we expect to see increased nutrient sequestration. So as plants are finding themselves, just to use sort of non-scientific terms, as they find themselves happier in soil with more dredge material, we hope that they will intake more nutrients because they like the addition of the dredge material, because when you add dredge material, you're adding the nutrients that are in there, but also you're changing the soil texture, you're changing a variety of different things. Most importantly, a previous study by Dr. Angelica Vasquez Ortega found that a professor at BGSU that is studying dredge material found that it greatly, there's a lot of calcium in dredge material, and calcium is an important nutrient in plants when it comes to root growth. So basically, as you increase the amount of dredge material, you increase the amount of calcium, and plants are like, oh, I love calcium. I'm going to, grow quicker. I'm going to be bigger than if I wasn't in soil that had a lot of calcium. So that's sort of a rough, these are the expectations we have for the hydrological and for the dredge material experiments. So now diving into what some may consider the more boring aspects of this presentation when it comes to experimental design. This is sort of how we set up the experiment, how we ran it, and sort of all of the design elements that go into that. So this was a mesocosm experiment, like I mentioned at the start of this presentation. We had plants in pots in 110-gallon cattle tanks. These cattle tanks, for the hydrological experiment, we altered the water level in there to mimic the different conditions. And then for the dredge material experiment, we had, because we had less species, we had them in a smaller mesocosm, but still. The mesocosm, in a way, is just a word for the cattle tank that we held all the species of plants in for each treatment. Like I said, we had plants inside cattle tanks. For the dredge material experiment, we mixed different ratios of dredge material into the soil that we used for both experiments. So for both experiments, we gathered soil from a nearby farm on the agricultural incubator property that we found that the soil most closely matched levels of organic matter, levels of phosphorus and nitrogen that we saw in real wetland soils, because it's very hard to get wetland soils because they don't want you to dig that up. So we decided to find soil that closely matched that, and a local organically farmed farm was willing to let us purchase a bunch of this soil. So that's the that's the soil that we used for both experiments. But for the dredge material experiment, we mixed dredge material into that in different ratios to hopefully understand how an increasing amount of dredge material amendment to the soil affects the plants. And again, we only mix dredge material into the soil for the dredge material experiment only. And then we hope to simulate different hydrological soil moisture conditions that we surveyed in wetlands, in the real wetlands in the field, by altering the water level in that cattle tank. So I'll have pictures that show that later too. And we only altered the water level for the hydrological or HABRI experiment only. and we kept the water level the same for the dredge material experiment. This is just to maintain, consistency and things. And then to answer this sort of the second question, we measured traits before they went in, during the on plants before they went into the pots, while they were in the pots during the experiment, and then after when we harvested everything. And then of course, it hasn't really happened yet, but we're going to take those nutrient samples as well. So this is a pictorial representation of our experimental design out at the Ecological Research Station at BGSU. We had this experiment outside because we tried to find greenhouse space, but it just wasn't large enough to be able to hold all of the tanks we would need for this experiment. So basically what the three different colors represent are the three different treatment levels for the hydrological experiment and I guess, and or the dredge material experiment with the three different ratios. But this picture just shows the randomization of those three different treatments in each block. And what a block consists of, an experimental block, is three tanks in a row. And then those three tanks would each have one of the treatment levels. And this is just a way to control against spatial bias or bias when it comes to the sun, because plants are the sun is a very critical source of obviously energy for photosynthesis in plants, but it just helped to control that and to prevent against biases when it comes to that. Now this is the actual tank setup for the Haber experiment or the hydrological experiment. You can see that we have 13 pots in here. We had one for each species, which it had 12 species, so we had 12 pots, one for each species, and then we had a 13th pot that was a control pot. This didn't have any plant in it, and this was just to understand what happens in these different hydrological conditions in soil without even a plant in it. So the plants, we gave a random number to each species of plant in each tank. So the order or the organization or the arrangement of plants, the species in each tank was randomized in each tank. And this is pretty much the exact same picture for the dredge material tank. We had 6 species, so one pot for each species, and then we had a 7th with just didn't have a plant in it. This served as a control pot. And again, the order of species in this tank was randomized as well. This picture demonstrates sort of roughly the 3 different water levels we had for the hydrological experiment. So we were trying to mimic with the first picture here on the leftmost section of this picture here, you see the blue rectangle is a demonstration of the water. So in the flood treatment or the high soil moisture or the high water level treatment, you can see that the water is very high. This was hoping to mimic the flooded conditions or very high soil moisture content conditions we see in wetlands, and this achieved the anoxia and all the stuff we were after. Then you can see in the normal sort of the control treatment or the medium soil moisture level, you can see that the water's halfway up the pot, so 15 centimeters. I forgot to mention, but all of the pots we used were 30 centimeters or 12 inches tall. So that's in lieu of not putting all the measurements on here for every single, I'll just tell you that now. So for the normal treatment, you can see the water level was at halfway up the pot. And then for the drought experiment, there was no water in the cattle tank at all for 4 weeks. So these are, there's sort of if you can imagine it, it's like a step down or a step up when it comes to wetness of soil. So those are what we hope to mimic and create in the Mesocosm experiment for the hydrological experiment. These 3 pictures are, actual pictures of the experiment going on. It's sectioned in the same way as the previous picture was. Well, the picture on the far left is flooded conditions. You can see plants look very happy. Wetland plants like the soil very wet, as you can imagine. So when conditions are flooded, they actually do pretty well. And then in the middle, you see the control or medium water level or the normal water level. You can see that plants are also doing very well in here. Surprise, surprise, wetland plants like it very wet. And then in the drought experiment, or the drought treatment, you can see that plants aren't doing very well. So, as you can expect, wetland plants don't like drought very well. But again, there are some species that are doing well. So why are they doing well as opposed to others? That's what we hope to answer. And these three pictures just demonstrate the visual differences when it comes to water level or treatment in the hydrological experiment. Now, leading into the dredge material experiment, this picture demonstrates the different ratios of dredge material to farm soil to dredge material inside the actual pot. So, again, the pots were the same in here. They're 30 centimeters tall. They were 12 inches tall, and they were 6 inches by 6 inches when it comes to length and width. And these pictures here demonstrate the different amount of dredge material that was mixed into the soil. So while the brown represents the farm soil and the purple represents the dredge material. So we had three different treatment levels of dredge material amendment, where one treatment was a 50-50 mix of dredge material to farm soil. One was an 80-20 mix of farm soil to dredge material, and the other was just 100% farm soil. So it serves sort of as a control. Um, it's important to note that for this experiment, we didn't just place dredge material and then farm soil on top like the picture demonstrates. We mix them all in with each other, so that way it's all mixed randomly within the pot. So it's not like this, but this just shows it much easier. In and then in the back, you can see the back of the pot, sort of. You can background of the picture. You can see that the water level for all three treatments was kept at that halfway up the pot, which mimics the control water level that we had in the hydrological experiment. This was just to keep consistency. Now diving into more of the methods, the actual hands-on work that went into setting up and taking down or setting up and running and taking down this project. We planted and raised over 700 plants. That's a lot of plants. Keep in mind, or just as a reminder, it wasn't just me working on this. We also had many undergraduates, almost 10 at a time, that helped work on this project. So you don't, while I did a lot of work, I did have a lot of help too. We measured multiple traits, anything from morphological traits, which is what I'll be focusing on in the results section of this presentation, and then we also measured physiological traits, and then we will measure nutrient content and nutrient content in the future. We then harvested all the plants, and then harvesting means we'll take each pot out of the tank and then separate the above and below ground and do all sorts of stuff to be able to make sure that we have all the different tissue types and roots in order, so that way we can do more stuff on them in the lab. And I won't touch on that right now because we don't have those results yet. And then again, once finished with all the processing in the lab, we will have not only the nutrient data, but all the other juicy data when it comes to root architecture and biomass differences. Real quick, I'm just going to show you guys some pictures that demonstrate the different things we had going on in this experiment, in both experiments. So this is a picture of overwintering in an environmental chamber at BGSU, and this is just to help plants to experience those winter conditions that they would in the field. And this is just to best mimic the natural seasonality of Ohio wetlands. You can see here, this is a student measuring morphology of a plant prior to it going into the experiment. So this plant was taken from a plug. So they weren't raised from seed. Most of them weren't, besides the annuals. We would take them from plugs and we put them in the pot, and then we would measure stuff as they went in to get a reading of initial biomass. This is a picture of a fully planted tank. While it looks very nice right now, this is at the beginning of the experiment. They didn't necessarily look like this the whole time. So that's sort of what we were getting after, is different responses to different treatments. This is a, albeit rough, picture of me taking chlorophyll readings of plants, and that's just to help us get an understanding of physiological measurements or how a plant is doing. How much chlorophyll does it have in its leaves, and therefore how much sort of photosynthesis is it doing? This is a picture of a plant that we take during harvesting. This is just the blue background is just to help us get a monochrome background to be able to understand the very, very visual differences in how plants look when it comes to each treatment. And yeah, and this is a picture here of more of the harvesting process. We would actually take the below ground material and split it in half and take a picture. This is just to get a very rough understanding visually of the root distribution in the soil, and then we would take samples further on. That would help us better understand that architecture and that soil, those soil characteristics better. And then finally, this is a picture just a few, probably just last week, of me taking scans of leaves to get leaf area. So that's another important trait that will help us to understand how plants are functioning in their environment in the treatments. I use this picture just as a not as an interruption, but just as a change of pace as I move into my results section. Um, I think, especially in grad school, there's a propensity in students to say, "Oh, this is so much work. Oh, it's so hard," and they just never see really how beautiful science is and how it helps us understand the natural world. This project was a lot of work to set up and to run and to harvest and to take down. And it was moments like this where I just look out at the experiment. This is the picture of the experiment while it's still, going on. I would say, this is all worth it. Science is all worth it. Not only is it beautiful, but it's just science is wonderful. The continuing understanding of the natural world and its processes is something that everyone should be aimed at achieving and understanding in some capacity. So, beautiful picture, but all in all, it shows a real, not just a graphical demonstration, a real picture of the experiment. Now diving into the results, I just have a quick prelude before I show you some cool graphs and stuff. We collected over 5,000 samples. So for a master's project, that's especially when it comes to plants, that's a lot of samples to process and stuff. And luckily enough, like I said before, I had a bunch of undergrad work, but still, it takes a lot of time to work through that. We just finished harvesting just a few weeks ago. And with all the busyness and the limited amount of people we're able to work at time and the limited amount of space, we just weren't able to process all of that when at the point of this recording. So there's going to be a lot of data that is going to be available next semester, which will be spring of 2026. So be on the lookout for any presentations by me or any of the Habri people then, because there will be a lot cooler data. Um, there is some data that is ready to analyze. There's a chlorophyll concentration index. This is a proxy for health or a proxy for photosynthesis. This is what I took. You see that picture of me taking that reading with the little clamper thing. This is that. Those just aren't ready to look through yet, and we don't have a real great understanding of that yet. So that'll be available again in spring of 2026. We took a bunch of water samples. These are actual physical water samples, sort of using a test tube, but also water samples using a YSI sonde that'll help us understand characteristics, physiochemical characteristics of the water and how different treatments are affecting the water, those characteristics. And then we also took morphological measurements. This was to help understand very differences in traits like height or stem count or leaf count between species. And this would be sort of a way to understand biomass and allocation and mass and all that stuff in just simple counting metrics. So we took these at the end of the experiment, and that's what I'll be, the morphological traits like stem count and leaf count are what I'll be focusing on in this presentation when it comes to the results. So, like I just said, morphology is a great means to predict biomass when but you either can't collect biomass because you're not allowed to harvest things in the field or in the greenhouse or in mesocosms, but it's also good for when it takes a lot of time to get all the biomass measurements. So, like I said before, our problem in our experiment is that biomass just isn't ready to analyze yet. We haven't weighed everything, and the things that we have weighed are different tissue types that we can't necessarily match up to a single species. So the solution to that is to use the morphological measurements that we have of each species um, each species in the experiment to be able to sort of predict biomass or just to see a basic response when it comes to the different treatments. The significance of being able to predict biomass and seeing a response is that plants will allocate biomass to different tissues in response to different conditions. So, in a variety of different conditions, plants may allocate more biomass to their leaves and they may have more leaves, or they may allocate more biomass to their stems and they'll have more stems. So, we hope the expectations of this are that we will be able to see responses in the number of leaves and the number of stems in the different treatments. So, like I said, the two different tissue types that I will use are stems and leaves. These are functionally, leaves exist on any, most plants that photosynthesize, and then stems also exist as well. So these are two things that are relatively easy to understand and also easy to count and quantify. So what I hope to show in these graphs is that differences in stem and leaf number between the treatments, so between the different hydrological conditions and between the different amounts of dredge material added, will mean a response. Will mean that that species of plant responded to the different conditions, and in turn it allocated more biomass, or it had different counts of stems and leaves as a response to the different conditions. Um, while these two measurements, stems and leaf count, are very important for predicting biomass, they're also vitally important for nutrient function. There's so many papers that I can show you that I studied that show that both stem and leaf count and stem and leaf size and height are all very important for how an organism functions as a nutrient filter, and also how it functions in its ecosystem, either as a competitor in terms of diversity when it comes to different um, species in the ecosystem. So now, this graph here is the results for the stem count for the hydrological experiment. Each one of these graphs here, each box or graph with with the species name above it, is its own graph for each species. The y-axis is the number of stems, and it's important to note that that y-axis scales for each graph. Some species have a lot more stems than others, so it's just a good. For me, it was visually, it's just better to show everything scaled, so that way you can see differences without, some being very small because they have 3 stems as opposed to one, and others have 100 as opposed to 300. So, what I want to highlight here is just some basic responses to the different treatments. And the different treatments in this are: the orange or the D is the drought treatment, the C or the blue is the control water level treatment, and the green or the H is the high water or the flooded condition. So I'm just gonna, so sort of this arrow down here indicates the increasing of wetness. So as you go from drought to high water on the x-axis, as you increase, go to the right, you have more, the soil is more wet. You have more wetness in the tank. So I'm just going to be showing you guys some real easy to see responses, either positively or negatively, to each treatment. You can see, for example, in this species up here on the top right, Elymus virginicus wild rye, that it had a very positive response when it comes to the number of stems. So it had more stems on average in the drought treatment than it did in the control. So this is interesting. Did this plant like the drought treatment better? That's what we hope to analyze more when we get more data. But this is just a basic response to see, oh, it did respond to the treatment. Maybe it likes that better. So we can investigate that further in the future. And then when it comes to Penthorum pseudoides, you can see that in the high water treatment, the green color, the green graph here, you can see that it had more stems on average than the control treatment. So maybe Penthorum pseudoides or ditch stonecrop really likes the high water treatment. And again, we'll investigate that further. Now some very negative responses. Juncus effusus, Shunoplectus, Scirpus, and Typha all responded very negatively to either treatment. So this means that maybe these species of plant like it just to be normal, and they don't like the high variety in changes in the hydrology or soil moisture in the soil. So, all in all, we hope that this. I will touch on the significance of this later on in this presentation. These are the leaf counts for the species. So these graphs are pretty much the exact same, where each sort of box or graph if it is its own species, and then the number of leaves is the y-axis. It scales again, and then the x-axis, the treatment, is the exact same. And I'm just going to be showing you guys some very easy to see responses when it comes to control compared to the treatment conditions. So, in these three species here had a very positive response in for Typha glauca or cattails that had a very positive response to high water, maybe not as much to the drought, but when it comes to leaves that had more leaves in the high water. So, um, maybe that's because there's a variety of reasons why that could be. It allocates more biomass because it doesn't want as much to be below ground because then it'll rot and stuff. And then that trend carries on of a positive response in the high water in Panthorum pseudoides, which is ditch stonecrop, and then Bidens sternua, which is nodding beggar's tick. So these three species in the high water treatment had more leaves on average. So that's a very positive response. That's easy to see in these graphs. That will hopefully, the reasoning behind that will hopefully be illuminated when we get more data in the future. Now, when it comes to negative responses, we have a lot of species again that responded very negatively to one or either treatment. Alisma, which is water plantain, Asclepias incarnata, which is swamp milkweed, Biden, and then Schonoplectus, which is soft stem bulrush, and Juncus effusus, which is um, just regular bulrush. They all responded negatively to both treatments. So again, these species might like the normal conditions in wetlands better than they do the very wet or the very dry. And then the two species that we mentioned earlier for positive responses had very negative responses when it comes to leaf count in the drought treatment. So, all in all, these responses to the hydrological treatments will help us to create a plan that I will touch on later on in this presentation. These are the results for the morphology for stem count for the dredge material experiment. So again, like the previous graphs, each box is its own species, and then the y-axis is the number of stems that scales for each graph, and then the x-axis this time is different though because as you move to the right in the x-axis, you increase the amount of dredge material added to the soil mix. So the orange in these graphs is 100% farm soil, 0% dredge material. The blue is 80% farm soil, 20% dredge material, and the green is 50-50 mix between the two. So I will again be highlighting some very easy to see responses. You can see that in Echinocloa that it had more stem in Echinocloa muricata or Kruskali, which is barnyard grass, had a very positive response in with the addition of dredge material. So this is sort of what we hope to see with increased biomass, increased establishment. Dredge material in specifically helped this species, but the amount of dredge material added affected the species differently. So that's what we hope to analyze in the future. But it's easy to see just a real positive response when it comes to control and dredge material added. And then for a very negative response, Carex vulpinoidea, which is fox sedge, did not respond very positively. It responded very negatively to the addition of dredge material when it comes to the number of stems. So maybe this species just doesn't function better when dredge material is added. And when you add dredge material, you should keep in mind that some species don't do good when it comes to the stem count. Now into the leaf count for the dredge material experiment. Again, each graph represents a different species. The y-axis is the number of leaves. Again, it scales for each graph, and the x-axis is the same, and the colors are all the same as well. So, just again highlighting some very easy to see positive and negative responses. These two species here, Biden sternua, nodding beggar's tick, and Schonoplectus, which is soft stem bulrush, responded very positively to the addition of dredge material. It varies based on the amount of dredge material added, but it's very illuminating to see, and it's a very preliminary understanding of how dredge material affects plants and a very elementary understanding, but still, it's very hopeful and it's sort of what we expected to see, where as you increase the dredge material added, you see an increase in biomass and the number of leaves. So for some species, it does increase biomass, and for others, it doesn't. So um, it is important to note, I forgot to touch on this earlier, that the reason why you're not seeing all six species in here or in a single graph for the number of leaves or all 12 species um, in the number of leaves and number of stems for the hydrological experiment is that when it comes to counting leaves and stems for things like grasses, it takes a lot of time and it's not a real good predictor of biomass. So when we took these measurements at the end of the experiment, we decided just to cut that out and to measure stem count for things that had a lot of leaves, and to measure stem and leaf count for things that had a very normal amount of stems and leaves. So that's why you're not seeing every single species on each of these graphs, is that we just either didn't count it or they're each, for something like Biden cernua, which pretty much has one stem. It's that number of stems is not going to change very much with in each treatment because it really only has commonly one stem, one main stem. So now some conclusions of what we hope to, overarching conclusions, responses by species that we saw in these two experiments. So flashing back to the slide, or a visualization of all 12 of these, the species in the hydrological experiment. These species that are outlined in green here had very positive responses to specifically the high water treatment in Biden's in Benthorum, and then a very positive response to the drought treatment in Elnus virginicus. So these species maybe like these conditions more, and maybe we could think about better about designing wetlands that cater to these plants more to create functional diversity. Whereas these three plants, Alisma, Juncus, and Schonoplectus, did not respond very positively to really either treatment. So, maybe when it comes to designing wetlands or seeding wetlands or planting wetlands in the future, you have to keep in mind that some species sort of just function better in normal conditions, and they won't function better as either in drastic changes within the year or different areas hydrologically within a wetland. These are the six. So just some real quick conclusions for the dredge material experiment. These three species here responded very positively to the addition of dredge material. So when you're planning, maybe in the future, when dredge material is used in creating wetlands, they can say that, oh, these three species like it a lot more. We should plant these species in this wetland in higher volume than we do other species because we know they're going to do very good. But again, we don't really have all the data when it comes to function as we're trying to answer it. We really just have biomass. So stay in tune for more data in the future. There's a lot of exciting data. But now flashing back to the conclusions, Carex vulpinoidea fox sedge responded very negatively. So again, you have to keep that in mind, maybe in the future when planting wetlands that have some dredge material added. So overall, I'd just like to give a quick summary of what these graphs mean, and again, what we hope to answer as more and more data comes in. So all in all, our focus is optimizing wetlands, making wetlands better as ecosystems, as diverse ecosystems, but also better as nutrient filters, because that's sort of the funding for building these wetlands comes from the need to make them function as nutrient filters. So that way we're not having all these harmful algal blooms and all this, these nutrients getting into the lake. So the hydrological experiment is all in all, when we get all of the data analyzed, including the nutrient data, all the biomass and all the traits, we hope to be able to create, or we expect to be able to create a climate smart planting guide. So, when you're creating a wetland and you've surveyed that area for a few years prior to it, the various hydrological conditions that it will be exposed to when it comes to, wetness, soil moisture, precipitation, and all the variation. You would know that. So using our data, the data that I'm gathering for my masters and the data that Habri is gathering overall, we hope to create, if you're designing a wetland that's it's gonna have areas that are very dry and areas that are very wet. Plant this species in the wet area, plant this species in the dry area. That's sort of what we hope to create. Or if you're going to have a wetland that varies a lot within the year or year to year when it comes to hydrology, plant these species because they do very good when the water level is or the hydrology is all over the place, or if you have a wetland that's a lot more stable, plant these species because they're going to do a lot better, um, you know, when it comes to more stable hydrology. And all in all, we sort of hope that the tendency is that we should just plant one species in wetlands because it's the best species at taking up nutrients. When in reality, that may not be the case when it comes to different plants in different hydrological conditions year to year, within the year, or within a wetland. You may need to you need to plant a variety of species because a variety of species, different species will function better differently. So that's what we hope to create is a better plan, a more efficient plan for design and planting of wetlands and restoration of wetlands in the future. So it's a very, very, very exciting prospect, and I'm so excited to be able to be a part of this and potentially have groundbreaking, revolutionary findings when it comes to wetland ecology and wetland restoration and wetland science. Now, with the dredge material amendment, this is sort of a different aspect where if we show that dredge material has a very positive effect on the establishment of plants, that may help to encourage the people, lobbyists or politicians to be able to pass laws that allow the use of dredge material in wetlands, and that just creates another use for it. That's eliminates the buildup of dredge material in these plants, and it also helps wetlands be better. It helps them function better as nutrient. If we show that it functions better as nutrient filters, that's another, that's more icing on the cake. And the whole aspect of optimizing wetlands, if you can't tell by the tone of my voice and the smile on my face, is something that I'm very excited about. And all the data that we're going to have, while it may not be ready now, it's going to be very exciting and very interesting to look at in the future. So keep in the lookout for any presentations that I may do in the future, or that anybody on the Habri team, whether it be Helen Michaels or Lauren Brown or anybody, does in the future of this project, because we may be able to have results that lead to better wetlands and lead to better design and lead to actual legislative change when it comes to dredge material that allows wetlands to function better in many folds. So this is not only just nutrient filtering, but it's also designing wetlands better when it comes to ecosystem variety of ecosystem services, but also habitat, ecosystem diversity. The vouching for native species and for using dredge material, and it's all just too much to share in one slide, but I hope you can say that, see my passion and excitement for it, and that this will result in some really cool findings and papers and manuscripts and guidelines in the future. So, as I talked and ranted on about the future plans, I hope to touch on this is some more specific stuff. Again, leading into the winter of 2025 and the spring of 2026, I hope to be able to analyze more of this data as it comes in and as it's processed. And I hope to also connect this to the field results. So we're also doing a very similar thing where we're taking biomass and stuff from the field. And that gets us the real actual what's going on there. And we can connect that to the Mesocosm results. And that leads to a more, integrative, all-encompassing approach to designing better wetlands. I also hope to connect this to all of the data that H2 Ohio captures. So H2 Ohio captures all sorts of things from bathymetry, so the topography of wetlands, to soil moisture, to soil water, to water content, to soil content, all sorts of stuff. So with that data, I hope to connect it to the data that we have to be able to, again, design better wetlands. That's groundbreaking. That's new stuff. But there again, with any master's project, you can only be so big. You can only study so much. So there will be a lot of unused data as the spring semester in 2026 ends. So I've actually, I'm a master's student right now in my second year. I'm intending to graduate in July, but I'm actually staying on for a PhD in the same program, working with the same people in the same sort of systems. So I'll be able to take all of that unused data, that I'm not using in my master's thesis and incorporate into a PhD. So um, I hope, selfishly, I'm very excited for this, but also for the sake of Ohio water, H2 Ohio, wetlands, Lake Erie, anything. I'm very excited for the prospects of this research, and it's not just me doing it; it's a lot of people. So with that, I'd like to acknowledge some very important players when it comes to these two projects. So, doctors Kevin McClooney, Helen Michaels, Steve Hovick, and Angelica Vasquez Ortega were all PIs on these two projects. So they all helped with coordination, with receiving funding, with designing the project, and helping to make sure everything is up to scientific snuff. And then there's three people: Ethan Glassman, Lauren Brown, Eliana Griffin, that were all important in coordinating the work that needs to be done and in designing and in data management and all this sort of stuff and organization. And then I am eternally thankful to all the undergraduates that worked for HABRI. There's unfortunately there's too many to name in this presentation, but if you're they know who they are and they know how important they are, and this project wouldn't even be able to get off the ground or even start or anything without all of their help. So I'm very thankful to them. I'm thankful to my friends and family that supported my journey along the way, even in the middle of the experiment when it seemed like it was, doomed for failure, and we ultimately achieved a lot of success. I'm thankful to them for standing by me and encouraging me and all the undergraduates the whole time. I'm also thankful to the BGSU biology faculty for assisting in various inputs on how to make the experiment better and how to optimize things and anything pest control wise too, and also for allowing me to walk through the building with noisy equipment and muddy gear. And then, most importantly, somewhat most importantly, I'd like to thank all of the funding organizations. Enough can't be said for these funding organizations. They're putting money out there to be able to fund great research and things that will make a lasting difference in this world. And to that, I'm eternally thankful to them for applying the funding to be able to have these two projects in the first place. And this is just of vital importance. And this sort of the funding of these projects will hopefully continue in the future. Um, and with that, again, if this was a presentation, I would have a section for questions from the audience. But if you do have any questions about this, this is my contact info. You can contact me at my email, or you can call or text me at my phone number. The best is probably email because it's sort of hard to relay long messages or questions over phone. I'd just like to thank everyone for watching this video. I know it's quite long, but there's a lot of stuff I'd like to touch on, and I hope this was very illuminating to what we have going on at BGSU and what we have going on in wetlands and the difference and the real lasting change we hope to make in Ohio and in wetlands worldwide. So with that, I am ending the video recording now, and I hope everyone has a wonderful time. And thank you so much again for listening.